So are we ready?
Good afternoon, sir. Here is Dr. Zai Kini.
Yeah. नमस्कार मॅडम.
नमस्कार सर. एक 5 मिनिटं वाट बघूया.
हो चालेल.
लोक बरेचसे, कारण कॉलेजमध्ये इतर सगळी कामं सुरू आहेत.
ठीक आहे मॅडम.
तो एक 5 मिनिटं.
म्हटलं रिझल्टचं काम तर
So, good afternoon, all of you. I am Dr. Varsha Tangde, coordinator of this workshop on behalf of Research Department of Dr. G.D. Paul Foundation, YMT Ayurveda Medical College and Hospital. I welcome all of you. First, I would like to welcome our respected guest speaker, Dr. Mukund Sabnis sir, and our Dean, Dr. Sanjeev Yadav sir, Vice Dean, Dr. Ninat Sathe sir, and all the teachers, PhD students, and PG students of this institute. Today, we are here for the online workshop of good manufacturing practices. First of all, we will have a brief introduction of this workshop. So, I would like to invite Dr. Zai Kini ma'am, Professor and Research Director of our institute, to introduce the workshop. So, welcome ma'am.
Namaskar. Myself, Dr. Zai Kiran Kini, welcomes all of you on this one-day workshop on good manufacturing practices as an organizer on the behalf of Dr. G.D. Paul Foundation's YMT Ayurvedic Medical College. Our G.D. Paul Foundation is leading in education, healthcare, research, Ayurveda authentic practices under the valuable guidance of our pioneer and chairman of our institute, that is respected Dr. G.D. Paul sir. We YMTMC is working continuously and dedicatedly for our vision and mission of innovation in teaching, research, and healthcare under the super dynamic leadership of our respected Dean, Dr. Sanjeev Yadav sir, and our respected Vice Dean, Dr. Ninat Sathe sir. We have our UG, PG, PhD, and fellowship courses, which are committed to inbuild the new or next generation of Ayurveda practitioner, researcher, as well Ayurveda entrepreneur. Today, as a part of our research and innovative teaching in healthcare, we are organizing today's workshop As a part of Ayurvedic practices, good manufacturing practices with ethical consideration is the main vertical. Our Rasashastra and Vaishudhya Kalpana department is ethically, religiously, and successfully working in this area under the leadership of Dr. Pargunde Madam. And now here we are trying our best to update our knowledge on good manufacturing practices under the precious guidance of Dr. Mukund Sabnis sir. Good manufacturing practices ensures that the products which are continuously or consistently produced and controlled according to quality standards. It helps to improve the production of goods by all means. Ultimately, it will reflect on patients' health and Ayurveda recognition in all over world. Being authentic Ayurveda practitioner, every Ayurveda graduate, every Ayurveda students, every Ayurveda researcher must know and understand about the standards of good manufacturing practices. So the person who is working sincerely since so many years and had set the benchmark in this area, that is Dr. Mukund Sabnis sir, is today with us to guide on this topic. Once again, I welcome Dr. Sabnis sir. I would like to request our research department workshop coordinator, Dr. Varsha Tangde Madam, to introduce sir. Tangde Madam, please.
Thank you, ma'am. We all are well familiar with our today's guest speaker. Still, I would like to introduce sir. So, Dr. Mukund Sabnis sir, MD Rasashastra, is a founder president of Jeevan Rekha Ayurved Chikisalaya and Research Center. He has worked extensively on standardization and stability of Ayurvedic medicines. Also developed a unique management for obesity, where more than 48,000 of patients have been benefited with the treatment. He has worked extensively on obesity and hyperlipidemic disorders and prepared herbal medicine.
ओके तो शायद नहीं आएंगे फिर गोधरा से.
He has also developed SOPs for treating PCOS and coronary artery disease patients and have formulated various medicines as per individual need. He also has a wide experience in Ayurvedic drug formulation and drug development and formulated more than 50 formulations which are marketed successfully. Sir has association with KLE Academy of Higher Education Research, Bharati Vidyapith University, Interactive Research Schools of Health Affairs, Pune, and also with Sri Jayendra Saraswati Ayurved College and Hospital, Chennai, and Yog Vidyadham, Nashik. Sir has received many awards in the field of research and drug formulations, like the Dhanvantari Award 2007 in appreciation of devotion to Nima. He has also received the Koratkar Puraskar of Vaidya Khadiwale Ayurved for research in Ayurved in 2011, Ayurved Mitra Puraskar from Arogyadham in 2018. He's a member of Sub-Advisory Committee CCRS New Delhi, also a member of Steering Committee of Interpathy Research, Maharashtra University of Health Sciences, Nashik. He has published many papers in the national and international journals, and also he is the author of books like Chemistry and Pharmacology of Ayurvedic Medicinal Plants, Concepts of Rasayan, and he has also delivered more than 100 speeches and talks on Ayurveda on All India radio and television channels, and we are well familiar with him with his many webinars and lectures regarding drug formulations. So We are eager to hear from you, sir. And I request sir to start the session. So please.
Hello,
Hello. Hope
Sabnis sir, you can start with the session.
Yeah,
Share
yeah.
the screen.
Hope
Yes.
I'm audible to you.
Yes, sir. You are
Okay.
well audible.
So, सर्वे सुखिनः सन्तु सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःख भाग् भवेत्. First of all, I thank to the committee and people of YMT College, Kharghar, to call me and for this webinar as a resource person and to listen my thoughts regarding good manufacturing practices. Actually speaking, good manufacturing practices, if we want to learn, you can just go on the web and just type good manufacturing practices in Ayurveda and you can have number of things before you. So what about then why this lecture is for? So I think today's lecture we will have some discussions. I don't want to point out any flaws, but we basically as an experts in Ayurveda and specifically coming from Rashastra Bhaishya Kalpana as expertise, we should have a recurrent and frequent discussions on with the rules which are been employed by the government of India in Drug and Cosmetic Act. Another thing that how we can utilize this rule to make our medicines up to a certain level. That is also important. And third thing is how to implement whatever the test they have mentioned, whatever the parameters they have given, how to actually interpret it in our day-to-day practice. Now, if you go through the Drug and Cosmetic Acts, it is said that those medicines, the act, it employs for those medicines which are kept on shelf, which are sold in the market, which have then FD, which have to go for a FDA approval, and then they are kept on the shelf on the market, and then there we have to apply for the good manufacturing practices. Another provision has been made in good manufacturing practices for Ayurveda. I will tell you the laws and which schedule it is there. But before starting that discussion, it has been also allowed for the Ayurveda practitioner, a BMS graduate, that he can prepare his own medicine for his own patient at his clinic. And majority of the people, that is the best part, because every physician, Ayurveda doctor feels that my patient should get a best quality of the medicine. And when we want to give the best quality of the medicine, we actually try our level best to deliver the best quality of the medicine at our clinics also. But these good manufacturing practices are also to be followed, though they are not mandatory, they are not compulsory, but they should be followed by each and every physician who is dispensing the medicine from their own clinic. If that is followed, then we can have a good quality of Ayurveda medicines. And basically, when it comes to Ayurveda, because the word good manufacturing practices has come from all over the world, and these good manufacturing practices was employed for Ayurveda also because there is a business involved into it. And when it comes to the business, that nobody should cheat, nobody should make changes into the formulation, and all these parameters were to be applied. So these good manufacturing practices, which were actually prepared all over the world for the contemporary medicine, they are also applied. That is the best part, that they are also applied for Ayurveda, and we also have our own parameters, that these are the parameters to bring such and such drugs into the market. And if we follow those parameters, definitely we can assure about the results of our medicine. That's very important. Unless and until we are keeping certain parameters regarding when there are many things which are to be visualized, which have to be kept under control of the manufacturer to prepare the medicines. So as we know that in Rasaratna Samuchaya, even in Charak Samhita also, there are specifications about Rasashala that I have given in the first slide. That where the raw material is to be kept, where the equipments are to be kept, where the furnaces or the putas are to be, in which direction they should be, where should be the place of the Rasashala. All the things are mentioned very, very clearly in Rasaratna Samuchaya. Even Charak has also mentioned in certain areas. So where should be the washing and cleaning? So cutting and chopping. So what should be the exact area of the total Reshastra? What it should not be near the drainage line or near the salvage and all these things. So these are all mentioned very clearly. Today also the same parameters are followed, that where should be the Rasashala or where should be our pharmacy regarding this geographical location. So when we talk about the good manufacturing practices in this definition, good manufacturing practice is that part of quality assurance which ensures that medicinal product has consistently produced and controlled to the quality standard appropriate to their intended use and as required by the marketing authorization or product specification. So marketing authorization is very, very important. What is the marketing authorization? And that there should be a reproducibility of the result and the reproducibility of the quality. And I think it is more and more, it should be stringent towards Ayurveda medication. It's very important, because if we think that, okay, I have to prepare a paracetamol tablet, my chemical structure of paracetamol is ready. And if I want to prepare the bulk paracetamol, I can prepare in bulk paracetamol within 15 days, 20 days, I can prepare tons of paracetamol. As we saw in COVID, that there was a high demand of paracetamol. You know that there was a WhatsApp message that Dolo 650 was sold like anything. And this manufacturing could be done within few days in the pharmaceutical companies. But if you see that, if that demand would be for Triphala Churn, if that demand would be for Chyawanprash like medications, then what would have happened? From where we would have brought this Amla, Hirda and Beda in that much amount of quantity? So there were chances to be what we can call as adulteration of the medicines. Malpractices could have done there, because we can't produce amla. If there is an order of amla of nearly 20 tons at a time, and if I have a capacity to procure 2 tons only, there will be a shortage of 18 tons from where I should procure. Then I can add some spurious or what we can substandard amla into my any product, maybe Triphala Churna or maybe Chyawanprash, for example. And that can have an impact on the quality standard of the product. And that is the reason why I feel that yes, GMP, that is good manufacturing practices, are to be employed very, very stringently, very strictly for Ayurveda medication. And if we are following it, definitely the belief about Ayurveda into the society, basically any science becomes popular due to the demand and supply chain. If there is more demand, there will be more popularity of the medicine. And if people start believing about the standardization of Ayurveda medicines, then definitely demand of Ayurveda medicines will increase. As demand will increase, there can be what we can call as substandard medicine can come out of it. There is a second very important thing why I tell that these good manufacturing practices are very important. When we go for the accreditation of our pharmacies in good manufacturing practices, maybe accreditation with national accreditation committees like maybe NABL and all these things. So they have one score that is called as a Z score. What they do, they take the samples prepared from our pharmacy and they analyze it. They take the samples of other 20 pharmacies which are accredited with them, and then they compare the results, analytical results. If the analytical results are very, very similar to other companies which are accredited, then we also get the accreditation of our certification for our pharmacies. So that Z score is very important. It means that the quality of all the Triphala Churna coming into the market should be one and the same, which can produce the same result. And that's the need, that we need a very, very stringent good manufacturing practice. Then there is again a different manufacturing practice that is called as a CGMP, which is not actually mandatory for Ayurveda medicines. But when we go through the good manufacturing practices into Drug and Cosmetic Acts, we can see certain parameters they have elaborated, they have told to perform, which again correlates with the current good manufacturing. So here the C stands for current. What do you mean by current? So C stands for current, reminding manufacturers that they must employ technologies and systems which are up to date in order to comply the regulations. So if we see that whatever the tests are to be applied, even Ayurveda pharmacopoeia also mentions about HPTLC, HPLC, LCMS, GCMS. So these are up-to-date parameters. These are the up-to-date instruments which are to be employed for this. It's very unfortunate as we say that okay, CGMP is not applied to Ayurveda, it is not mandatory to Ayurveda. We miss it. We perfectly miss it that it is not applied, so we don't need it. But if we apply these type of CGMP parameters to our products, definitely our product can stand at a certain level. They can stand at an international level. And that's why, though it is not mandatory, we should see that, because I am not going to just elaborate what is the GMP which has been mentioned in Drug and Cosmetic Acts. You can, everybody can read it. But what are the things which we have to discuss? So we should see that our products which are getting prepared into the pharmacy, they are matching with CGMP or not. It is not mandatory. Just going for ash value or water soluble extractive or acid soluble ash, all these things, these are very superficial parameters. And these parameters in the era of 2022, these parameters are really outdated parameters. They can't tell about the perfect quality of the drug. For example, I'll take one example of same example of amla. We will take that what should be the aging of the amla that should be used in Triphala Churna or Chyawanprash, or any other certain herbs. We can tell what should be, for example, I want to take Arjun bark. What should be the age of that Arjun tree which has a optimum amount of the active principles which can elaborate a perfect result? Ash value or water soluble extractives or simple TLC can't elaborate it. We have to go for a very specific tests. And to go for these specific tests, no doubt though it is not mandatory now, and then I'm telling it is not mandatory, but we should see that we should compete with the CGMP parameters which has been told in the Drug and Cosmetic Acts. Okay, so so what are the regulations? So the Drug and Cosmetic Act 1940, Section 33, and it was first elaborated. Then it was amended, that Drugs and Cosmetic Act Rules 1945, and another amendment was done into Drug and Cosmetic Acts 1999, and the Schedule T was prepared, which is specifically, Schedule T is specifically for Ayurveda drugs and Ayurveda pharmacies, their testings, raw material testing. So all that has been very clearly mentioned in Schedule T of Drug and Cosmetic Acts of Rules 1945 and thereafter 1999. So what is the need of GMP? Why it is needed? So to assure the public, now I am again a big question mark, not only public, to assure Ayurveda fraternity also. I am practicing, though I am also in academics, but I am practicing Ayurveda in my clinic since 28-30 years. And there is a big question mark when we start our practice, or when any student or PG student starts the practice, and when he doesn't get the results, the first doubt is about question. Today also, the first doubt is about Ayurveda and our drugs. So to assure not only the public, to assure self, to have a self-confidence within a doctor, within the physician, that Ayurvedic drugs are safe. Very important. And not only safe, Ayurvedic drugs are effective. You can assure yourself only at that point only that yes, Ayurvedic drugs are safe and effective when they are properly standardized. And that's the important need of GMP. Now again and again I'm telling you, to assure ourselves, to assure all the Ayurveda fraternity first, because there are lot of Ayurveda students still studying, lot of Ayurveda graduates who have passed out. Those don't believe in Ayurveda. That is a fact actually. And to have a good impact of this GMP can make them confident about Ayurvedic drug safety and Ayurvedic drug effectiveness, both the things. Second, to ensure that raw material used in the manufacturing of the drug are authentic, of prescribed quality, and are free from contamination. Again, very important. Now, when we talk about contamination and when we compare with modern medicine or synthetic drugs, they're all synthetic drugs. And then these drugs, they are less prone for fungal and bacterial infection, whereas our herbal medicines, they are more prone for multiple infections, maybe viral, maybe yeast infection, maybe fungal infection, maybe more bacterial load, maybe load from the soil. So all that contamination, the medicine what we are using as raw material, they should be free of contamination. So GMPs for that, so that as compared to this. So the manufacturing process has been prescribed to maintain the standards. Adequate quality control measures are adopted and the manufactured drug release for sale is of acceptable quality. Now these questions that they have a quality control and they are accepted quality, how it is decided that they are acceptable quality? Then what we do when we buy some extracts or when we buy some chemical drugs, for example, we buy aspirin, we buy paracetamol or any other synthetic medicine, allopathic medicine, when we buy it or when we buy some extracts nowadays, we get a certificate of analysis, so we call it COA. So when we demand a quote of the raw material, we ask the provider, the supplier, that whether you are going to give a COA, and the provider say yes, we are going to give a certificate of analysis of the drug. Now see, if you go into the market, and just I'll tell you some examples. Famous, what I met with, a famous example that if you want to buy a B12 in as a raw material, everybody knows that there is a B12 deficiency nowadays and many people take B12. And if you want to go to buy B12 into bulk, the cost of B12 in the bulk market ranges from 2 lakhs per kg to 36 to 40 lakhs per kg, and every company will give you a certificate of analysis. But at what level they are giving the certificate of analysis? Not at the level of CGMP. They may not be giving fingerprint. The person who is selling B12 at the cost of 2 lakhs rupees per kg, they will not give the fingerprint. They will not give the peaks. But the person who is selling at a 40 lakhs rupees kg, he will give a fingerprint, he will give a peak of B12, everything he will give. So this is the difference between the certificate of analysis. That what type of certificate of analysis I am getting out of the product, out of the raw material. That is very, very important when we are going for the manufacturing of the products. That's why though GMP is applied, we should try to compete with the CGMP when we are into the good manufacturing practices. That's very important. So what is the difference between GMP and CGMP? Both CGMP and GMP are put into place to certify quality assurance of the product. GMP assures that the product is exactly what the product claims to be and what it was created by the standard guidelines set known as GMP. However, a little difference between CGMP and GMP, the C in the CGMP, which denotes the current GMP, are stated they were finished in more current manner. Means whatever the analytical methods are updated in the world, not only in India, maybe in US, maybe in UK, they are mentioned in US pharmacopoeia, maybe in UK, maybe in BP British pharmacopoeia. All those parameters they are applied on while selecting the raw material. They are applied when we are processing the raw material, making the medicine out of it, and they are applied for the finished good also. So this is the difference between a GMP and CGMP. So though it is not mandatory, again I'm telling that you should compete, you should try to go for CGMP for our products. So another difference between CGMP and GMP is the cost. No doubt the cost of analysis will be very high, but the product will be sold at that higher level only. If you see in the market that you can get a aspirin tablet at a cost of 15 paisa, you can get an aspirin tablet at a cost of 5 rupees also. Then we said that what is the difference? Difference is between the quality, between the analysis what they have thought about. Maybe both the aspirin may be same, or there may be some difference. What kind of difference there may be? Okay, the label claim is of 15 milligram. They might have added 10 milligram. That can be the difference. Quality wise, they may be same, but the analytical methods, they were at a very, very higher level, which has made them more and more assured about their product, selling that at a very higher cost. So this is because of the new technology is expensive when compared to the GMP goods. CGMP goods remarkably more testing to prove the accuracy of it, and these more testing are every time beneficial for the people, for the society, and for the manufacturer also. So it has to undergo newer and more in-depth testing and requirements that FDA GMP, so that the certification is accurate and proven to be effective. So a normal GMP, as I told you about the Arjun bark, we don't know the age, that what amount of tannins will be developed, or what amount of the active principles will be developed into the Arjun bark, or what should be the age of the avla when we are plucking it. So that can be analyzed very, very perfectly with the help of CGMP, and this CGMP will make the manufacturer, the seller, the buyer, and the society assured about the result and safety of the products. That's very important. So when we go for the standardization of when we go for the processing of the raw materials, so standardization of the raw material is a very, very important, and that has been added into the Ayurveda pharmacopoeia with chemical profiling. It's very important that standardization of raw material with chemical profile, I have taken as it is from the Ayurveda pharmacopoeia, these slides. So standardization of raw material with the chemical profile, then SOP of extract if used, because nowadays many companies, we are using extract 10-15 years into our practices. So SOP of extract is very important. If I am buying some extracts and how the extract is prepared, we know that sanskaro hi gunantara dhanam. That's very important. As the sanskar changes, the pharmacological activity totally changes. So how the extract is prepared, whether the extract is spray dried, whether it is vacuum distilled, or whether any other, or what we call solvent extraction extracts. So what type of extract? Because what we see, okay, I want ashwagandha extracts, but which extract you want? Whether you want a complete water soluble extract, or you want a what we can call as water and alcohol extract, or you want a complete alcohol extract, or you want a chloroform extract. What type of extract we are using, depending on that, the total pharmacology changes. So when you are buying an extract as an institute or as a manufacturer, we should know the SOP of extract, that how the extract is prepared. We should go there, see the manufacturing process, because as the process changes, the pharmacological activity of the extract changes. Secondly, at what concentrations the extracts are prepared, means whether it is to 1:8, 1:5, 1:10, the concentration also, where as the concentration varies, the pharmacological values, the effects also varies. So this SOP of extract is very, very important, that a manufacturer should know to decide the dose of the product, to decide the pharmacology of the product. So these are very, very important thing, and this extract are to be standardized with the chemical profile. So the standardization of the extract, SOP of extract, then SOP of the formulation, that how I'm going to formulate out of those extracts or certain raw material. Forget about only extract, but about any raw material, how I'm going to formulate it. And when it comes to formulation, how I am bringing the product. Whether, for example, I want to take gulwel as a raw material, whether I am buying gulwel as a fresh gulwel or as a dry gulwel. Then whether if I am buying a dry gulwel, how was it that made dry? That is a SOP of the raw material. That whether it was a dry, means shade dry, or whether it was a sun dry. Because shade dry will have a different chemical profile, and the sun dry will have a different chemical profile. So these type of things should be noted very keenly. So standardization, formulation, finished product, and shelf life studies. So one by one we are going to see this. So this is a schematic diagram of this good manufacturing practices. So SOP of the formulation of any formulation, your SOP should be very, very clear. For example, if I am preparing Chavanprash, whether I am preparing as per the Sharandhar part, or whether I am preparing it with as per the charak part, pharmacology may differ, and it differs. So SOP of the formulation should be mentioned on the label and should be noted very keenly when we are preparing the medicine. Then standardization of the formulation, then process standardization is very important here. How we are processing, at what temperature we are heating, at what temperature we are cooling, all these things should be noted very clearly. So process standardization is very important. And then finished product and shelf life studies. Again, shelf life studies are very, very important when we are specifically with Ayurvedic medication, because they can get oxidized, they can get infected very fast. So shelf life again depends on the packing procedure actually. How airtight packing you are, which type of packing material we are using into it. So that type of depends on the shelf life studies. Then the biological screening, then safety and toxicity studies, and then preservation, packing, storage, and dispatch. So this all the things comes under a good manufacturing practices. So when it is a newly approved therapeutic agent, again we have to see that what type of molecule we are preparing, whether it has been mentioned in the selected given 50 books of Ayurveda or not, whether we have a reference, whether we have a textual reference, or we have a research paper for that, that is to be studied first. Then drug like small molecules from the herbal medicines. For example, I want to add Haridra into certain product, but I want its high potency. So what I do, I will take curcumin, nanocurcumin, and my label claim will not be nanocurcumin. My label claim will be Haridra only. But I will add a nanocurcumin to improve its activity. Whether it will work? No. Basically, I have to mention it very clearly. I have to give a reference from the research papers or from the text that yes, I am using nanocurcumin, and the process of preparing nanocurcumin is mentioned, and this is the process. So this way we have to go for. If I want to add, now andrographolite is also coming from active chemical from Andrographis paniculata. So these andrographolites, how are they prepared? That should be mentioned in my SOP, and that should be very clearly declared that this was the andrographolite or curcumin, this was processed that way, and then I am adding into my formulation, and that is the reference. So that will decide about the drug small molecule from the herbal medicine, then going for the conventional pharmacology, then biological data sets, disease date sets, and then safety, efficacy, then the pharmacokinetics and pharmacodynamics are to be elaborated for the new drug. Mind it well, again, the pharmacokinetics and the pharmacodynamics of all the dosage form from Ayurveda itself are totally different. Their indications are different. Means the pharmacology of Shatavari Swaras, pharmacology of Shatavari Churna, pharmacology of Shatavari Kalpa, or pharmacology of any product made up of Shatavari may be totally different, and the pharmacokinetics may be different. So as it comes in modern medicine, okay, if we don't have injectable diclofenac sodium, we can give tablet or we can give suppositories. It will not work in Ayurveda. Everywhere the pharmacology changes as the processing changes, so that should be kept in mind. So we have certain fixed things as per modern medicine in our mind, but these fixed things are not applicable in Ayurveda. They are totally different from each other. So that pharmacodynamics and pharmacokinetics should be, at least logically, at least they should be elaborated very, very clearly, and that can give a very good impact for the product development and for the quality assurance and the safety assurance of the product. So regarding herbal extract, I have spoke that water soluble components, what are the heat liable components. For example, if I have some herb, for example, we'll take Piper longum, or we can take shunthi, or we can take trikatu, any ingredient from trikatu which has good volatile oil, and I have prepared extracts with a spray dye method. So all the volatile oil has been already gone, already oxidized. That type of extract will not be of use of mine. I have to go for the vacuum distillation method to collect all that volatile oil or the active principles which are very heat liable, and that then I have to prepare extract out of it. So when I'm going for herbal extract, the method of preparation of the extract should be focused, should be studied, elaborating the pharmacology of that herbal extract. Because if the active principle is the volatile oil and it is heat liable, it is not heat stable, and then if I am heating it by the spray drying method, I will not get the results. So I can't assure about the quality and I can't assure about the safety of the product. So these are the things, that whether how much fat soluble components are there, how much heat resistant components are there, what are the water soluble components, what are the heat liable components. So these, and what are the techniques to prepare these methods of the extract, that are to be known to the manufacturer very well. Just buying the extract as it is and adding it to our products will not suffice. That will not give a quality assurance, though you will pass the pharmacological test for the purpose of the legality to sell into the market, but the quality will not give the expected results, and that will lose the confidence into the society. That is the biggest loss actually. What if we are not following GMP? What will be the things? We, first of all, we will lose the confidence from ourselves itself, from the Ayurveda fraternity people, and second, we will lose confidence from the society. So basically people will feel, "Arey, it's a Ayurveda medicine, it will not work." They expect this, they start taking medicine as it will not work. So if we want to cut down these what we call as bad patches about Ayurveda, then definitely we have to go for a proper GMP for Ayurveda medicines. Then coming for the shelf life or date of expiry of the medicines. So the date of expiry of Ayurveda, Siddha and Yunani. So basically date, Charandhar has very clearly loved. Gunahinam bhaved varsham urdham tadrupa aushadam. That all the slok from Charandhar Sanhita. Masadvaya tatha churna hina viryatvam apnuyat. Hinatvam gutikale ho labete vatsarat. So all the expiry dates and about the shelf lives, Ayurveda has already thought for it. So how to put into on the paper now? The date of expiry of Ayurveda, Siddha or Unani medication shall be conspicuously displayed on the label of Ayurvedic, Siddha or Unani medicine, as the case may be, and after the said date of expiry, no medicine shall be marketed, sold, distributed, or consumable. So that is very clear. Everybody, since our childhood, we have made a habit to see the expiry dates of the product, and if the expiry date have passed, we throw out the medicines. That Ayurveda has also, Sharandhar has also clearly elaborated things, provided that this rule shall apply to Ayurveda, Siddha and Unani medicine seeking license or renewal or license for manufacturing after the date of notification of these rules. When the notification has been made, that is not important. Okay, as per legal aspects, it is important, but as per the consumer, it is not important. For consumer, the efficacy of the medicine is very important. So if we want to restore the efficacy of the medicine, definitely shelf life of each and every product should be identified very correctly, and that should be mentioned on the label claim. So what the Drug and Cosmetic Act says, or what the good manufacturing practices says, that so they have elaborated the shelf life date of expiry with effect from the date of manufacturing. When it comes to Anjan, it is Anjan made from Kashtavadi 1 year, Anjan made from Kashtavadi along with Rasa Uparas Bhasma 2 years, Arka 1 year, Asavarishta 10 years. So Avleha, Khanda, Paka, Guda 3 years, Churna and Kwatha Churna. Now again, there is a big question in my mind. It is just out of discussion. Nothing about this Drug and Cosmetic Acts mentioned in the rules. But Kwatha Churna is, there is no entity as a Kwatha Churna. Churna is Churna. That whatever the Charak in the Panchavid Kashaya Kalpana they have mentioned, or Sharandhar in the Panchavid Kashaya Kalpana they have mentioned, that Churna is Churna. There is no entity as a Kwatha Churna. So you should not believe in such thing that there is a Kwatha Churna. Kwatha Churna is not there. So Churna, Lepa Churna, Dantamanjan Churna. So 2 years is the expiry date. Dhupam, 2 years is the expiry date. The Dravak, Lavanak, Shara, they have mentioned 5 years. Grita, 2 years. Gugulu, 5 years. Gutika Vatika, again 5 years. Then Gutika Vatika only with the Kashtaushadi 5 years, and 3 years, and only with the Rasa and Uparas, that is a 10 years. So Asavo, Dhatavo, Rasaha. So Asava and Dhatava and Rasaha, they have a long expiry, that they have mentioned that the Asava Arishta and Uparas, that Gutika Vatikam only from Rasa and Uparas Bhasma, that is a 10 years. That's very nice. But we have to follow it. We have to print it on the label. We have to sell it. Only then we can sell when we have a printed manufacturing date and expiry date. It's very clear. Now, again, we should study ourselves also that really whether these ras and uparas are not getting oxidized when they are packed properly or when they are not packed properly. Whether these churna, whether they have a stability. I have read one paper from some traditional medicine from Japan, and the Glycyrrhiza glabra, which was kept thousands of years back, was that paper has occurred in Indian drugs in I think 1995, 1996 thing, and that Glycyrrhiza glabra was still very safe, and that was having a highest part of glycyrrhizic acid, which is active component. So it can stay even more than that if the packing procedures are very good. So we should again start working on the shelf life of the drugs. We should work in the labs for that, and then we can decide actual what are the shelf life. This is for the legal entity, it's okay. But then also, as a Ayurveda practitioner coming from Rashastra Vaisheshya Kalpana, we should see that actually what is the fact, that how much they can be stable, and after what time they can become unstable, or they can catch infections with them. So this again and again I'm telling, that is again related with the how we are packing this material. So if it comes into contact with the atmosphere or the oxygen in the atmosphere, they can get oxidized, they can get infected, and the shelf life can deteriorate very badly. So similarly, for Nasa Bindu, for the Kupi Pakwa, for the ointments, it has been mentioned like 3 years. Any application, any local application that they have mentioned 3 years. Mandura Loho, that is 10 years. Nagabhasma, Vangabhasma, again they have reduced up to 5 years. Netrabindu 1 year, Parpati 10 years. So there is a big list that you can go to the net, and then you can see that what are the expiry date or the shelf life they have mentioned, and as per that we have to make a label claim to sell the product into the market. But we should also see that what is the actual seen into the, that how much is the actual stability, how much is the actual accelerated stability regarding all these things. So what the law says, that for long-term studies, frequency of testing should be sufficient to establish the stability profile of the drug. For a drug with a proposed shelf life of at 12 months, there should be 3-time testings. When we are producing it, and then in the 6th month and in the 12th month. If it is for the annual, if it is more than 3 years, for more than 1 year, then frequent checking after 6 months, we should again make an analysis for our products, and that too with CGMP if possible. That too with CGMP to assure the product that yes, it's good into the market. At the accelerated storage condition, a minimum of 3-time point, including the initial and final point 0, 3, and 6 months from a 6-month study is recommended. Reduced design, that is matrixing or bracketing, where the testing frequency is reduced or certain factor combination are not tested at all, can be applied if justified. Only if justified. Otherwise, we should go for every 3 to 6 months testing when we are deciding a shelf life of a particular product. Now, evaluation, the purpose of stability is to establish the base on testing a minimum of at least 3 batches of the drug, a retest period applicable to all future batches for the drug substance or a shelf life, and label storage instruction applicable for all future batches of the drug product manufacturing packed under similar circumstances. And Ayurvedic drug can be considered if no significant change. Now see, it's very important that Ayurvedic, any drug, not only Ayurvedic, but any drug can be told as safe when the no significant change. And now you can see the definition. What do you mean by no significant change? See now, plus or minus 20% change from the initial assay value if the drug is analyzed for its marker, or plus or minus 15% change for the initial assay value if the drug is analyzed for its active compound. Can you imagine that? What do you mean by 20% plus or minus change? 20% इकडे किंवा तिकडे. Total range 40%. Can we assure the stability of the drug when there is 20% on the positive side or the 20% on the negative side? The physiochemical parameters like moisture, ash, particle size shall not vary beyond 25% of the initial value. We have to think for it. Okay, for legal purpose, it's 100%. I don't want to comment anything on it. But as an academic person, as a scientific person, we have to think that whether the moisture, if the moisture content of my Shatavari Churna goes 25% above the initial thing, it can catch fast infection because it is already hygroscopic. Or any moisture percentage which is catched by any herbal powder or churna, they can catch infection, and then we can't assure. But legality says it's 25%. It's okay for 25%. But we as Ayurved people should understand these things and see that it should not deviate anything from 1 to 2%. Because we nowadays have a very excellent packing procedures, very excellent packing materials. With the help of these packing materials and these packing procedures, definitely we can maintain a perfect moisture value from the first day to the third year of that or second year of that powder. Particle size can be maintained very nicely. So these are very important things which are to be again rethought about this significant change value. So these significant change should be not much as per the science, but as per the law they are telling about 25%. It's okay. So failure to meet the acceptance criteria for appearance, that is physical attributes, and functionality test, that is color, phase separation, caking and hardness, then we can say that yes, if there is more caking, because some churnal which are more hygroscopic like Shatavari, they can cake and they can hard very fast. Tablets, they can deteriorate, they can become more fragile. So these are many things which can be just affected by moisture. So these things are to be kept in mind when we are packing the things and where we are deciding the shelf life of the products. Then talking about the raw material standardization. So passport data of plant material, definitely we should know that what are the test parameters which has come in the annexure 3 of the raw material standardization. So passport data of plant material, part of plant use, botanical description, adulteration reported in literature, and substitution reported in literature, that is allowed. But if I'm getting ashwagandha from Maharashtra, if I'm getting ashwagandha from MP, and if I'm getting ashwagandha from Kerala, and that is cultivated, the amount of ethanolides differ a lot. So that we should know the geographical condition from where the ashwagandha is brought is very important to standardize the raw material. When it was, when the cultivation was made, when the yield was done, and how it was dried, all these passport data of raw material is very important, specifically in Ayurveda. So when we are collecting raw, what happens in modern medicine? No, we can compare it. In modern medicine, there are some bulk drug manufacturers and there are some dosage drug manufacturers. So these dosage drug manufacturers, they buy the product from the bulk drug manufacturers and they have a certificate of analysis. Now the drug here in regarding Ayurveda, our drug bulk manufacturers may be farmers or maybe collectors of the herbs from the jungles. They are totally illiterate people. So we have to keep a keen watch on them, that how it was collected, how it was harvested, how it was dried. All this passport data is very important when it comes to Ayurveda. Then foreign matter, if any, and acids insoluble ash and total ash, very important things. Now you see that we have a parameter that your total ash should be of certain herb should be 8 to 10%. Our herb has passed that test. Okay. Our acid insoluble ash may be certain 7 to 8% passed. But what is the ratio of total ash to acid insoluble ash? We have to interpret it. Because why we are doing acid insoluble ash? Acid insoluble ash is very important regarding the herbs actually. That two herbs which are connected with the soil. For example, Nagarmutha. If you take an example of Nagarmutha and you tell a farmer to bring Nagarmutha to your clinic, he will bring Nagarmutha along with the soil. So how much amount of silica and silicates it has, how much amount of earth it has, how much amount of mud it has, that is decided by the acid insoluble ash. So this thing about all the dashamulas, acid insoluble ash is very important. This total ash to acid insoluble ash, this should be matched very correctly. Moisture content at 105, this should be matched, because if I am using little bit more moist herbs, then it will catch fungal infection in the finished products. So these parameters which has been elaborated in the annexure 3 are very important. our interpretation regarding this test should be also very important. As a pharmacist or as a Ayurvedic expert we are working, so our interpretation is also very important. So again, now I told that though the annexures have included CGMP, they have not made it a mandatory. So they have written that TLC, HPTLC, GLC, HPLC, these are all higher parameters, but they have also mentioned about TLC. TLC is outdated. But if we are able to use all these things, HPTLC, GLC or HPLC, then you can just, you are competing with the CGMP requirements. So assays are to be performed about the total tannins, about the alkaloids, about the resins, but only these assays are not sufficient. So assays are to be prepared. Test for heavy metal toxicity, lead, cadmium, mercury, arsenic. So where this test is to be applied? If I am applying certain these type of test of heavy metals for the rasaushadis or the rasakalpas, means herbo mineral compounds, already I am putting some mercury intentionally. I am adding lead intentionally. So what about the heavy metal toxicity? So our interpretation regarding these things are very important. Then pesticide residues, no doubt, if we are actually, if you go through the Kalpasthana of Charak has clearly mentioned that you should not take herbs which are cultivated. They should go grow in the jungles and then we should pluck it. But nowadays, because of the more demand, we are cultivating the medicines. So about the pesticides, so again it comes about the good agricultural practices. Ayurveda is not limited only for what we can say about the good manufacturing practices. As the drugs or our raw material is coming from the fields, is coming from the nature, good agricultural practices are also very important. Good mineralogical practices about the collection of mineral is also very important when we are preparing these or when we are testing these raw materials. So test for specific pathogens, that is E. coli or any pathogenic bacteria, that is okay. Aflatoxin detection is very, very important. A simple herb, I'll tell you. Kadunimba, Azartica indica. So this Kadunimba, it has a very, very great affinity towards aflatoxin. Aflatoxin is that toxin from the fungus which is highly carcinogenic. Now simple Kadunimba leaves, if we are adding to certain our churnas, for example anti-diabetic or any other thing, and if the moisture content of this Kadunimba has been on the higher side, then it can catch aflatoxin very fast, and it can prove, if the patient is taking it in a regular manner, it can prove to be highly toxic. So these type of things are very, very important. Aflatoxin retention is very, very important in our area. So again, coming to the shelf life is also a very important thing. Is very, very important thing when we are talking about the raw materials. Even raw material shelf life is also very important. Now when we talk about the shelf life, every time we think about the finished product shelf life, we have to also think about the shelf life of the raw material also. That when the raw material has occurred in my company, when the raw material I have brought, what was the stage at that time, and then when I am using it. So the shelf life of raw material is also to be defined and is to be noted very clearly. Then about the plant extract, I have explained all these things. So these are the certain parameters regarding the plant extracts. So residual solvents are very, very important. Bulk density of the plant extract is very important. Alcohol soluble extractives, water soluble extractives are very important. Now acid insoluble ash doesn't play any role in the extract because they are all extract. So the mud or the earth will not come into the acid soluble extract. But here water soluble extract, when the company is claiming that they are giving extracts, the extractive values of water should be at the highest, and if that is not meeting, we can't elect a pharmacological response out of it. So these parameters are very, very important. Then the shelf life of extract also, remember well, the shelf life of powder and the shelf life of extract is totally different. I'll tell you, though we are using haridra as an antibiotic in the patients, haridra is the herb which get infected very fast itself, and that's why the shelf life of haridra extract and shelf life of haridra as a raw material is very, very important. Similarly with neem, similarly with many herbs. Regarding the pesticides, we were moving into the farms for the collection of the herbs. We saw a field where ginger was grown in tons, and there was a big, like ginger godowns were there, and ginger was kept for drying into the sun. Then just we inquired with the farmer that, okay, you are so lucky that you are having so good quality of ginger, and you might be using it as a medicine very now and then in your food, and as a medicine you might. So the farmer told us that no, 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 it's not like that. We never eat this ginger. Then we were surprised to listen that we never use this ginger. I asked him that why don't you use this ginger? So he told us that ginger is that herb that which needs highest pesticides, and we have to get a good amount of yield, we have to add high pesticides into ginger. And we, when we are consuming ginger, how many of us think about the pesticides when we are using it in our medicines also? So the amount of pesticides which are used into such type of herbs which are cultivated, that is also very important. See the experience when we move in the farms. I was shocked to hear that ginger needs lot of pesticides to grow, and these farmers, because here the good agricultural practices are very important. So when we are buying the product, we should have an agreement with the farmers. We should visit the farm. We should see what are the conditions of the soil, how they are grown, how they are cultivated, how much pesticides, whether they are using controlled pesticides or not. All these are to be thought. All these are, this is to be documented. Then and then only we can go for a good manufacturing practice. So these good manufacturing practices regarding then, as we saw about the extracts, we saw about the churna, and we saw about many things. So these good manufacturing practices are to be taught in a very small manner. Then only we can, our finished good can come out with a good manufacturing practices. Then coming for our churna yoga, that is churna, the same things are applied everywhere. Avleha, leha, modaka. So the same things, that what is the specific gravity, what is the total acidity, what is water soluble, extractive, alcohol soluble. Then physical appearance is also very important. Make it a point that physical appearance. Now just I'll tell you that what is the color. When we think about the physical appearance, okay, I saw that specific churna was of certain light yellow color. So we mentioned light yellow color. Just if we buy a colorimeter which cost 20-25,000 rupees or much less than that, and if we make the wavelength, that at what wavelength we are, what is the wavelength of this color, if we are able to measure it and write it down, we can apply the organoleptic character into the machine, and we can make it on the paper that okay, the color is light green and having the absorbance of such and such nanometer of the light. So simple it is. But if we go for that, definitely we can see the changes according to the time. Yes, after 6 months there is a slight change in the color. So the slight change in the color shows that there is a slight change in the physiochemical parameters. And as per the pharmacology, as the physiochemical parameters of the drug changes, pharmacology changes. That's a simple principle. As the chemical property, as the physical property of the drug or of the herb changes, the pharmacology changes. So going to this principle, a simple color order can be brought on the paper in the numerical value, and this can be again identified while looking for its quality and looking for its shelf life also. Again here, when we go for the standardization of the churnas, again 40 to 60 mg, we can mention 40 to 60 mg because Kwatha churna has not been mentioned especially into our granthas. There is no special category of the churna that there is a Bharad churna. Bharad churna is also never mentioned in our text. So from where the Kwatha churna and Bharad churna has come into this, these things, I don't know. But yes, okay, it has been mentioned in Drug and Cosmetic Acts. We have to follow. But we can just mention churna up to 40 to 60 mg. It can have a different stability timing, different shelf life. Particle size of 80 to 100 mesh, it can have a different pharmacological activity, or it can have a different shelf life also. because as the particle size changes, the rate of uptake from the gut changes. It increases. Less the particle size, more the rate of absorption of the drug. The pharmacokinetic changes, pharmacodynamics changes. So these are very, very small things, but we are not concerned with this frequently. We should have a concern with these very small things. Then we can have a very nice assurity and surety, or assurity and the safety of the drug can be guaranteed very nicely. Now coming to the pushti and bhasma parameters. Now description, same color, odor, taste, pH. Now when we see that, when we tell that it is a metallic preparation, bhasmas are totally metallic preparation, and whatever the organic material has been occurred there, it has been totally burned into carbon. And what quality of carbon it is a different story again, but it has totally burned into carbon. Now if we look into the aqueous extract of that bhasma, can we get the aqueous extract of the bhasma? It's very, very difficult to have aqueous extract of the bhasma, but here it is given, we can mention it. But as a scientist, as a science, we have to think a lot into it, that okay, how can bhasma have an extract? Similarly, a total ash of bhasma, because when I go when we go through the research papers or the dissertations of MD and PhD students, they mention ash value of the bhasmas, and the labs also give the ash values of the bhasmas. Bhasma itself is a ash. How can be there a ash value of a bhasma? And the ash value is expected up to 99.5%, or 99.9% can be ash value of the bhasma. This bhasma itself is a ash. So we have to rethink about all these things. And when we go for the HPLC and GLC for all these parameters, it can't be possible. Similarly, now again, if you come to the standardization requirement, if you see, they have mentioned about IR, they have mentioned about XRF, they have mentioned about XPS, they have mentioned about XRD, they have mentioned about electron microscopy. So they have mentioned about what we call as AFM. So they have mentioned about AA, atomic absorption. So these higher parameters have already mentioned in the GMP. It is just a point that we should start to apply it very correctly. If we are trying to apply, if we try to apply it very correctly, there will be definitely reproducibility of the results. There will be definitely assurity about the shelf life, assurity about the efficacy and the safety of the drug. So I'll just I urge that everybody should go for this CGMP, which is already mentioned but not made mandatory for the Ayurvedic drugs. Then regarding the parameters which has been mentioned about the tailas and ghritas, now all these are very correct, that viscosity, iodine value, saponification value, then unsaponifical matters. Here again, very more. But one parameter, when we are preparing taila or ghritas, the most important thing which is a problem with the pharmacy is about the filtration. So here we can add one more parameter from our side, that is a particles detection into the tailas or ghrutas. And if they are detected, what are the size and how can they? So we can use that type of mesh to filter it. So this is a very routine problem with the pharmacies, that the particles of the kalka dravyas or the churna dravyas which are used while processing, they come into the taila or ghruta. So these particles should not be seen. So this type of parameters we can add from our side into the pharmacopoeia, or sorry, into the good manufacturing practices regarding these dosage form. Now when it comes to gutika and vatika, so the disintegration time is that time that the tablet should disintegrate into the abdomen to have a very good uptake. Now if you see the what we can call as the abdominal transit time, or the abdominal emptying time, gastric emptying time, that is accurately that is called as a gastric emptying time. So gastric emptying time is 20 to 25 minutes. So within that your tablet should be disintegrated completely. Now we have a one more benefit that from this legal thing, that not more than 35 minutes except gugulu. Are the Googles any special medicines? If I have Amrita Google, if I have a Mahayogaraj Google, if I have a Yogaraj Google, if I have a Punarnavadi Google, So if these Googles are not getting disintegrated, so what is the normal parameter? They have more than 35 minutes except Google. So Google can have any time of disintegration time. Will it work? So let the law be like that. I don't want to debate on this, but at least as a entrepreneur or as an academician, we should see that okay, my Google tablets, though Google is not advised to be converted into tablets by Charak. He has never advised it. But then also, if we want to prepare Google tablets and bring into the market, we should see that their disintegration time should be below 25. Otherwise, they will not work. So all these things, forget about the legality, but we should see that our medicines should be more and more effective. So that those interpretation as a pharmacy owner or as a manufacturer, we should see these parameters are doing very correctly or not. Then all other parameters we have talked a lot on it, so I'll not waste time into this. Yes. Now we have a Indian pharmacopoeia. Now, when we go to the Varti, Netravarti, and all these things, they have said that the essays can be taken from IP, the essays can be taken from British pharmacopoeia, from the United States pharmacopoeia, or Ayurvedic pharmacopoeia. See that now, and it has been evident that there are nearly 40 to 45 herbs in Indian pharmacopoeia. There are many herbs now which are occurring in USP and BP. So we have to follow these books also, these pharmacopoeial standards also, because now Ayurveda is not limited up to India. If I want to sell my capsule or any tablets outside India, I have to follow their guidelines. And I have to see that what essays has been mentioned in BP or USP or European pharmacopoeia also. So these guidelines, if I am into the export, I have to use CGMP definitely, and I have to go for the all these IP and see the very wisely they have added here BP, USP, API, and IP. These pharmacopoeial essays are to be followed very correctly if we want to be in the exports of Ayurvedic medicine, and that we have to follow because we have to follow the guidelines which has been made by their pharmacopoeial standards. Coming to Panchavidha Kashaya Kalpana, as I told in the initial lecture only, that yes, the medicine which are sold that are to be undergo, that has to be undergo good manufacturing practices. It should not be like that. Even Vaidyas, if they are preparing their own medicines, for example, many people prepare Chavanprash or many prashes, many people prepare. We prepare churnas, we prepare tablets. So all those things are to undergo the good manufacturing practices, and they have to get it standardized from some other labs. So no tests for them, no parameters for them, and no guideline from them. Where Panchavidha Kashaya Kalpana is the basic thing for Ayurvedic practitioners. So we have to see that we have to apply the parameters for the preparations what we are preparing in our clinic also, apply the parameters for the assurity and for the surety of the results. Then coming to Schedule E1 of the Annexure 4 for from the Drug and Cosmetic Acts, there are certain toxic drugs, and these toxic drugs, I think the list should be have more things, that Aifen, Arka, Bhallatak, Bhang, Danti, Gomut, Datura. So whatever has been mentioned as a Vishavarga, they have mentioned it as a Vishavarga. I'll give an example. That Jatifal, Mystica fragrance. If a person consumes 4 to 5 Mystica fragrance, there can be a lethal activity on the body. Lethal things will be there on the body. So all such herbs which in a higher dose can prove toxic, if at a higher dose can prove toxic, then they are to be thought in a very different angle, and then that they are to be added into the formulation, though they are not enlisted into this list. So this is again a very, very important thing. So you should know Schedule E from Annexure 4. But apart from Schedule E, we should also see that whatever the medicines I am using, whether those medicines they are having high sedative activity or any other lethal activity in higher doses, that should be also taken into consideration regarding the safety of the medicines. Then coming to the very interesting part, that is the Schedule T of the good manufacturing practices pertaining only to Ayurvedic drugs. So Government of India notified Schedule T wide GSR number 560E on the 23rd June 2000 under Rule 157 of Drug and Cosmetic Rules, and further amended good manufacturing practices in March 2003. These rules became mandatory to all new Ayurveda, Siddha and Unani drug manufacturing units from 23rd June 2000 and for existing units from 2002 respectively. So Schedule T specify the requirements of the factory premises and hygienic condition. The main aim of introducing Schedule T was to maintain a uniform standard of the hygiene for the manufacturer and compliance to good manufacturing practices in mandatory for all the manufacturers. So a majority of the time, all the Ayurvedic manufacturers, that is a good part, that they are following Schedule T rules. So we can just look what are the Schedule T rules. So the raw material used in the manufacturing of the drug are authentic or prescribed quality and are free from contamination. Every time we should see that contamination is the biggest risk for Ayurveda. Infection is the biggest risk for Ayurveda drugs. So that contamination is to be thought for. Manufacturing process has been prescribed to maintain the standards. Adequate quality control measures are adopted. For what quality controls? For raw material quality controls, raw material shelf life, raw material storage, where you are storing it, whether you are storing it in the jute thaila or whether you are storing it in a stainless steel container, how much airtight it is, whether you are storing it in the form of powders or whether you are storing in the form of herbs as it is or as a kanda or as it is, what are the storing means in which form you are storing, that is to be seen, that is to be observed very keenly, and the moisture contents, because more the moisture, more the contamination to the herbs. So to achieve the objective listed above, each licensee shall evolve methodologically and producer for following the prescribed process of manufacture of the drug, which should be documented as a manual and kept for reference and inspection. This is very, very important. So whatever we are doing into the manufacturing unit or in our unit in the clinics, we should have the best of the inventory. First of all, our inventory should be online. This is not mandatory as per the but these good manufacturing practices can come to CGMP. It should be online. It should tell us that what are the storage conditions, whether the temperature is changing or not. Inventory should tell everything. Under IMCC Act of 1970, registered Vaidyas, Siddhas and Hakims who prepare medicine on their own to dispense to their patient and not selling such drug in the market are exempted. That's a good part. They are exempted purview of good manufacturing practices. But still I say that though we are exempted from to dispense the medicine from our clinic, still we should practice good manufacturing practices when we are dispensing the medicine also. That is the important part to get noted. So what are the part out of it of the Schedule T? That is part one, that is factory premises. So it should have adequate space, receiving and storing raw material, manufacturing process areas, quality control section, finished goods store, office, and rejected good and drug stores should be there. Then location surrounding the factory building for manufacture of Ayurveda, Siddha and Unani medicine shall be so situated and shall have such construction as to avoid the contamination from open sewerage, drain, public level. For example, if you have a godown, how is the godown plastered or if it is not plastered? Whether you have a paint over it or whether you have a complete vitrified tiles on it, it makes a lot of difference for the storage of raw material. So these standards are to be maintained when we are storing the raw material or when we are storing the finished product also. If we are storing the raw materials in a godown where it is not at all plastered or where the flooring is also not good, then the chances of contamination to the raw material is very high. That we have to monitor very correctly. So these are the general requirements of the raw material storing. So regarding the buildings, so buildings should be clean, tidy, just I explained. It should be have good ventilation, it should have good light, because whenever it happens in the godowns, when the light goes, when the electricity offs, then there is total darkness. It should not happen. It should have a proper ventilation, these godowns, because when there is lack of sunlight, again there are more chances of infection. So compatibility with other manufacturing operations that may be carried out in the same and adjacent premises. Adequate providing with working space to allow orderly and logical placement of the equipment and material to avoid risk of mix-up between different drug or components. So labeling is very important. How we are labeling the drugs in both means the label should be in English. When we are in Maharashtra, it should be in Marathi also. It should be in the botanical name also. So some or other person can't identify the botanical name. He will identify the regional name or the English name of that herbs. So label should be in a proper way, in a big letterings or big fonts. The label should be. designed, constructed and maintained for prevent entry of insects and rodents, that we have discussed. The wall of the room in which the manufacturing operations are carried out shall be impervious to and be capable of being kept clean. You should be able to clean the walls very frequently. A simple paint or non-plastered building wall can't be cleaned. So it should be with the vitrified tiles or the glazed tiles should be there on the wall, on the flooring of the godowns to store the raw material or the finished product. Provided with proper drainage system in the processing area, the sanitary fittings and the electrical fixtures in the manufacturing area shall be proper and safe. That's very, very important. If there is a big godown and the switch of the electricity is at a far point, long from the door, it is of no use. It can cause any hazard. So the electrical fixtures will be just adjacent to your door. Similarly, if there is a fire in the building, so the fire extinguishers should be at a hand reach into the godown or into the or in the manufacturing area. These are the things which are to be followed. Furnace bhatti section should be covered with tin roof and proper window. Here I want to give an idea to the Ayurveda manufacturers, very important. We every time think when we are into the herbo-mineral preparation or in the preparation of basmas, and when they are marketed and coming into the market, consumed by people, people have a concern that yes, this medicine has a heavy metal and this may be toxic and this may not be toxic, whatever it may be. But when we are manufacturing it and the fumes coming out of the bhatti or out of the furnace, they are also too dangerous than the consumption of the medicine. And that should be focused very, very keenly and very important, because mercury vapors are more dangerous than the consumption of parpati and kajali. You should make it a point. These mercury vapors are to be properly trapped, and the amount of mercury yield I am getting out of the maybe mercury shodhan or ashtavidha sanskar and all these things, how much mercury I am getting and how much the mercury is lost in the vapors, that much toxicity we are facing when we are preparing these type of herbo-mineral preparation, and that should be addressed very correctly, what I feel. So there should be a fire safety measures. So your fumes should be, whenever we are preparing herbo-mineral components, your fumes should be properly delivered out of, and at a very high height they should be delivered. So drying space should be very, very open, and they should be free from the insects, dust and flies. Water supply, the water used in the manufacture shall be pure of potable quality. Then disposable of the water should be very proper. Containers cleaning, very important. I'll tell you one example. I had been to one of the biggest unit of India preparing vaccine, and the way they accelerated their procedures. So they used to prepare vaccine into a very big drums initially, and then they realized that for every batch they have to clean the drum. And cleaning was a very, very tedious part of the company to prepare more vaccines more fast about this COVID-19. So what they did, they prepared one such material which is completely disposed after preparing one batch. So there was no problem with the cleaning of the instruments. So cleaning of the instruments after every batch or after different dosage form is very, very important. If you want to excel the speeds of the preparation, that your vessels should be proper and very easy to clean. So in factories where operation involving the use of containers such as glass bottles, vials and jars are conducted, there shall be adequate arrangement separated from the manufacturing process for washing, cleaning and drying of such containers. It's very important. Second very important thing, that whenever, for example, we have ball mills or we have end runners or we have tablet preparing medicine, what is the material from where the machines are made? If they are made from cast iron, definitely the lead and the iron will come from the machines to our product. So see that nowadays the machines are made up of complete stainless steel. Means what we call as there is a different grade of stainless steel used for machineries in the pharma industry. That grade should also be used for Ayurvedic medicine, because though we are maintaining a proper parameters for the heavy metals, and if our machineries are made up of different type of metals which can leach very badly into our products, our product will fail when we are doing testing at a finished product level. And then we will not be able to identify from where the lead is coming from, where the iron is coming, or from other metals are coming into my products. So the quality of these machines, the material used for preparing these machines, the metals used for preparing these machines should be of the stainless steel grade. Then raw material of metallic origin, it should be collected very perfectly, that we have talked about. Raw material of mineral origin, raw material of animal source, very important regarding basically cow's urine, honey, milk. Standardization is very tough, and that should be followed very stringently, because these are all perishable. Animal sources material are all perishable, so they should be standardized, they should be stored and utilized first on the first come first basis. Fresh herbs, very important, that when we are preparing some bhavan, some swaras, or making bhavana out of it, so these fresh juices should be utilized on the same day. They can't be stored. So that standardization is to be made into GMP. Dry herbs of or plant parts, how they are stored, excipients, volatile oil, perfumes and flavors, plant concentrate, extracts and exudate resins. So these storage conditions should be very, very perfect if we want to. And storage not only about the finished product, but the storage of raw material also should be focused very nicely. So storage should have proper ventilation and shall be free from dampness, should provide independent adequate space for storage of different type of materials, such as raw material packing, packing material and finished product. Everywhere the storage facility should be totally different. Now we have talked about the raw material. Again, just as a one of the query, that shelf life of raw material, in the last I have shown that shelf life of raw material is very important, and shelf life of raw material is very leastly focused, that when I have brought the raw material, in which stage I have brought, whether it was wet or whether it was dry, whether it was a finished powder as my raw material, what type of raw material I am buying, how I am storing it, and what about the raw shelf life of that raw material, that should be identified. So frequent test, as we are testing the shelf life of end products, the shelf life of raw material should also be tested to follow the good manufacturing practices. And if the raw material is approved or rejected, that way it is, we have to dispose it. If it is rejected, we have to clearly dispose it. If it is approved, then we have to take it for manufacturing on the first come first use basis. So that is very, very important regarding the raw material. All the raw material shall be sampled and got tested either by in-house Ayurveda Siddha and Unani experts, or by the laboratories approved by what we call as a PTL, public testing laboratories. The rejected raw material should be removed from the raw material store and should be kept in separate room, and procedure of first in first out, that should be maintained very meticulously. So packaging material, all packaging materials such as bottles, jars, capsule, etc. shall be stored properly. All containers and closures shall be adequately cleaned and right from packing the products. So these are to be followed very correctly. Similarly with the finished product, regarding the means, they have to go for the reverse calculation, that what is the demand from the market, that's how we have to produce it. We can't produce it, is not a non-edible product that we can just stuck it into our godowns and as per the demand then we can supply it. No, as per the sub demand we have to prepare our batches, and then we have to store it into the proper place, that is called as a quarantine area. After the quality control laboratory and the experts have checked the correctness of the fitness of the finished good with reference to its packaging, labeling as well as finished product quality as prescribed, then it will be moved to the approved finished goods stock area. So once the raw materials, finished goods are prepared, they are first kept at one place, that is called as quarantine, and when it is approved, then it is moved toward the approved finished goods stock area, and then it is dispatched for the distribution at a various location. Then the working space, the manufacturing area should provide adequate space or orderly placement of equipments and material used in any of the operation for which these employed, so as to facilitate easy and safe working and minimize or to eliminate any risk of mix up between different drugs. So working space should be abundant. So they have specified about the working place, that slide I'm going to show you. Health, clothing, sanitation and hygiene of the worker, that is also big concern regarding Ayurvedic drugs. So if your worker is having some skin problem, some infection, they can more easily contaminate the herbs, basically fresh herbs. They can more easily contaminate. So these fresh herbs contamination is to be stopped, and we have to check the hygiene of the people. We have to check the clothing of the people, whether they are changing their clothes every day, whether the company has provided the dresses for them which are changed every day into the pharmaceutical company. They are using the mask, they are using the caps over their head, because the hairs may be spoiling the finished product, also maybe spoiling the, while processing they can fall on the in the medicine also. So these things are to be taken care very nicely. Then batch manufacturing records. The license shall maintain batch manufacturing record. The licensee shall maintain batch manufacturing record of each batch of Ayurvedic Siddha and Unani drug manufactured, irrespective of the type of product manufactured. Manufacturing records are required to provide an account of the list of raw material and their quantities obtained from the store, tests conducted during the various stage of the manufacture, like physiochemical test and chemical test, as may be necessary or indicated in the approved book of Unani or Ayurveda Siddha Unani mentioned in the first schedule of the Drug and Cosmetic Act 1940. So these tests may include any in-house or pharmacopoeia test adopted by the manufacturer in the raw material or in the process material and in the finish. So these rules and regulations, they mainly focus on all the three: raw material standardization, process standardization, and finish standardization. The manufacturer has to first declare that these are the tests I am going to conduct on this particular product. They have to declare it first, and then they have to rectify it frequently. They have to go for these tests. Process standardization means when we are processing some material is taken out at a certain stage, again it is to be investigated, and then we can move to a finished product standardization. So these things are to be keenly observed. Then distribution of the records of sale and distribution of each batch, because many a times we see that cars are called back because of the manufacturing fault. We see that, okay, certain company has called that batch of the car back because they know that to whom they have delivered the car. Here it is very difficult. If we don't have a record of sale and distribution of each batch of Ayurveda Siddha or Unani drug, it will be very difficult that if we found that some mistake has been done or some problem has occurred into the processing and the batch was not prepared correctly, we can't call back the things. So these records are very important. Certain category of Ayurvedic Siddha and Unani medicine like Bhasma, Rasa, Kupi Pakwa, Parpati, Sindura, all these medicine do not have expiry date. In contrast, their efficiency increases with the passage of time. Hence, record need to be maintained up to 5 years of exhausting of the stock. It's very important. Means it is the responsibility of the manufacturer that till the expiry date, whether the medicine has gone into the market and distributed into the market, but till then, till the expiry of their end manufacturing drug, they have to maintain that sample into their premises and keep a watch on that shelf life. It's very important. So record of market complaint should also be there. So if there are certain record complaints from the market, they should be maintained very perfectly. So manufacturer shall maintain a register to record all reports of market complaint receiving regarding the products sold in the market. The manufacturer shall enter all data receiving on such market complaint, investigation carried out of by the manufacturer regarding the complaint, as well as any corrective action initiated to prevent the recurrence of such market complaint. So if some complaint is there, what action the manufacturer has taken against the complaint, that should also be recorded very nicely, and the register is to be maintained for all these things. Then regarding the quality control, we have talked, we are speaking regarding the quality control only. There should be 150 square feet area for quality control section. The identification of raw drugs, reference book and reference sample should be maintained. Manufacturing records should be maintained for the various process. To verify the finished product, controlled sample of finished product of each batch will be kept for 3 years at least. That's mandatory. That once I have given a batch and my batch is exhausted, I have given to the distributor and distributor had distributed to the medical stores. But till the 3 years of till the expiry of that batch, manufacturer has to keep that sample and keep on testing the samples. To supervise and monitor adequacy of the condition under which raw material, semi-finished product and finished products are stored, keep record in establishing shelf life and storage requirement of the drugs, and manufacturers who are manufacturing patent proprietary Ayurveda Siddha and Unani medicine shall provide their own specification control reference. That's very, very important, because majority of the products which are coming into the market are patent and they are proprietary medicines. And about the proprietary medicines, where there are no direct references about the formulation from the text, they have to create their own parameters, and those parameters are to be declared, and those declared parameters are to be noted, and the manufacturer has to investigate all these medicines on a regular basis and keep a record out of it. Now the record of specific method and procedure of preparation, that is bhavna, mardan and puta, that is a big question mark now. And the record of every process carried out by the manufacturer shall be maintained. I'll give you an example. When we were studying at a postgraduate level, we had a very good department of Rashastra and Vaishya Kalpana. We were working under respected Saurikar sir, and we took to prepare Gandhak Rasayan. Now see, Gandhak Rasayan has 96 bhavnas, and we started preparing it. And one day we found that it was quite impossible to give a color to Gandhak Rasayan or give a bhavna to Gandhak Rasayan, because every eight bhavna there is one bhavna of Adrak Swaras into Gandhak Rasayan. So 96 plus 12, all these calculations we have made, and it was taking 1.5 years to prepare Gandhak Rasayan at our level at that time. It may be in 1994. So we found that after a certain period, it was converting into plastic sulfur. So how these people mention that we are preparing Gandhak Rasayan and we have done all these bhavnas? No. Today we don't have any procedure, any parameter to identify whether the prepared formulation has been given certain amount of puta or certain amount of bhavna. Now the definition of bhavnas are also different. That's why the procedure is very important. Dravena yavata samyak sarvam dravyam prutam bhavet. Means that type of decoction or swaras we have to add into the color, that all the material is getting poured into it. So that is one. Diva diva atape shushkam ratro ratro nivasayit. That is again a different definition of bhavna. So what type of bhavna you have given or you are intending to give, that should be clearly noted and into the processing, and that should be seen that we are using the same procedure. Now, for example, if you take an example of Arogyavardhini, where we have to give 7 bhavnas of Kadunimba Swaras. Now, if I give 3 bhavnas of Kadunimba Swaras and bring the batch out to for the market, how I am able to identify whether the bhavnas which are given, whether they are 3 or 7? So it is our duty, coming from the scientific community, to develop certain tests to identify that how much bhavnas have been given on that particular drug, or how much putas has been undergone for a particular bhasmas. So this is a big question again. We have to believe on the manufacturers, and then we have to see that whether the bhavnas are given in a proper manner or not. So this is again a big question among all of us. The standards for identify purity and strength has given in respective pharmacopoeias of Ayurveda, Siddha and Unani system of medicine published by Government of India shall be compiled with. All raw materials will be monitored for fungal, bacterial contamination with a view to minimize such contamination. Now see, government has not told that you should maintain a microbiology lab, but they are telling that you should see the bacterial and fungal contamination. From where to see? Whether if the infrastructure don't have a microbiology lab, you can't see it. So these are the things where we have to work on. These are the things which actually indirectly that is mandatory to us to maintain all such parameters. So one person with Ayurveda Siddha Unani qualification recognized under Schedule 2 of Indian Medicine Central Council Act 1970, 2 other person, one each with the bachelor qualification in botany, chemistry, pharmacy, could be one part of the contractual basis. So one biochemist and one Ayurveda doctor or Ayurveda physician, they are the integral part of the contract for the manufacturing of the process. The manufacturing unit shall be a quality control section as explained under section 35(2). Alternatively, these quality control provision will be made by getting testing etc. from a recognized laboratory in Ayurveda under Rule 160A of the Drug and Cosmetic Act. The manufacturing company will maintain all the record of the various tests got done from outside recognized laboratory. So if you don't have this facility, you can go for the public testing laboratories and get the test done. But it is very important to get these tests done. So what are the list of equipments they have advised? So there are, as per the dosage form, the list of equipments are very big list. So grinder, disintegrator, pulverizer, powder mixer, sieves, shifter, big list is there. But again, I am telling that the quality of the material of the machines, metal used for equipments, they are to be standardized. They are to be such metals which will not react with the herbal material or the decoctions of the or the asawaristas. They will not react with them, and those type of machineries are to be used. So yes, now very important part, that Ayurveda Siddha Unani drugs section, the guidelines for issue of license with respect to Ayurveda Siddha or Unani drugs. Ayurveda Siddha or Unani drugs includes all medicine intended for internal or external use for in the diagnosis, treatment or prevention of disease or disorder in human beings or animals, and manufactured exclusively in accordance with the formulae described in the authoritative books of Ayurvedic, Siddha and Yunan Tibbi system of medicine as specified in the first schedule. It's very clear and we are following it very strictly. Nowadays, the formulations are coming Panis ginseng with ashwagandha formulation, label Ayurved medicine. Saw palmetto with gokshur, label Ayurved medicine. St. John wort along with Ayurved medicine. Ginkgo biloba along with Brahmi and Shankapushpi. Now, here the point comes about the first slides, that whether we have gone for the herb to herb interactions, whether we have gone for the pharmacokinetics and pharmacodynamic studies for all such type of formulations which are not used in the formularies or the 50 books which has been mentioned in the schedule of section 1A of Drug and Cosmetic Acts under Rule 158B. So these are the very important things which we as a Ayurveda physician or a doctor or as Ayurveda fraternity, we should think over that, and then we should apply the things very particularly. So so these are the issue of the license of the medicine with respect to Ayurvedic, Siddha and Unani medicine. So if you have a reference from the Ayurvedic books, there is a, about the ingredients, indications, safety studies are not required. So if the ingredients are not in the text, if they are as per the text, or the indications are new, then the evidence is required, means the research papers are required. If the patent and proprietary medicines, ingredients are as per the text, indication are as per the textual and their rational, safety studies are not required. So they have made a provision. Or if there is a new drug is coming, no new formulations are coming, definitely pilot studies are important, research papers are important, and unless and until there is evidence, we can't bring such type of combinations into the market. So we should observe all these things which has been elaborated very nicely, and luckily we people are implementing it as a good manufacturing practice, not only in Maharashtra but all over India we are implementing. No doubt these rules and regulations are given by the central government. There are again state governments which are having the power to implement it. So as per the states, there are different rules and regulations. A minor difference is there into the regulations. I will just explain one difference into the regulation and then I will stop this lecture. See, if you want to manufacture a capsule coming out of extract of Boswellia serrata, you can't manufacture in Maharashtra. You have to go for a Gujarat FDA. Gujarat FDA promotes and helps or permits to fill Boswellia serrata extract into a capsule and market it. But Maharashtra FDA doesn't allow. I don't know the reason behind that, but these are the differences what I have put forth. So here I end with this lecture, and come just one slide about the good agricultural practices, that harvest location, growth condition, stage of the plant growth at harvest, harvesting time, collection, washing, drying and storage condition. We have to educate the farmers regarding using of the pesticide and insecticides, then quality specification, namely foreign matter, total ash and acid insoluble ash. These are very important when it comes to the relation of the raw material, not with the finished products. But acid insoluble ash, there means where there is a contact of the plant material with the earth, with the mud or with the soil. Here the acid insoluble ash and total ash, their ratios, they play a very important role. Heavy metal contamination, if the harvesting is along the roadside, it can catch lead and all these things. So heavy metal contamination is very important with the raw material. Chromatographic fingerprinting profile is very important with phytochemical reference marker, assay for bioactive or phytochemical components. So these good agricultural practices are also to be performed. Means for Ayurveda manufacturer, there is an additional responsibility that they have to keep a watch on the farmers with them, with whom they have made a contract for their yield. So that is again a important responsibility on the Ayurveda manufacturers regarding the good agricultural practices, whether the farmers are following it or not. So here I end with the lecture. Thank you. Thank you very much.
Okay. Thank you.
Thank you very much, sir.
Thank you, sir. Thank you very much for your brilliant session today. And it was really a much needed session and a valuable training for all of us. So, audience, if anyone has any queries, please feel free to ask.
If anyone wants to ask the question, they first kindly introduce themselves and then they can ask anything. Okay? कुणाला काही प्रश्न विचारायचे आहेत का? वर्षा मॅडम, माझा आवाज आहे, येतोय ना ऐकू?
हो, येतोय मॅडम.
So if you have some questions, you can just mail me or you can just WhatsApp me. You can take.
Sir, I request you to kindly put your mail ID in the chat box. जरा चॅट बॉक्स मध्ये तुमचा मेल आयडी टाकाल तर मग सगळ्याकडे रिसर्च यूज म्हणजे पीजी आणि पीएचडी स्टुडंट्स पण आहेत, द्रव्यगुणचे सुद्धा आहेत, तर त्यांना हेल्पफुल होईल. जर काय असेल त्वरित तर ते तुम्हाला मेल करतील.
Yes, I will put the things in the chat box.
Thank you so much, sir. Varsha madam, you can continue.
Okay. So if there are no queries, or if you want to ask afterwards, you can ask your queries to sir. So for today, I would like to thank sir for your such a experienced and personable presentation today. And as we are towards the end of our session, I would like to thank our guest speaker, Dr. Mukund Sabnis, sir, for taking the time out for this session. And we all appreciate your expertise and also expect your collaboration in future also. Then I would like to thank our chairman of this institute, respected Dr. G.D. Poe, sir, for always being our inspiration. And I would like to thank our Dean, Dr. Sanjeev Yadav, sir, and our Vice Dean, Dr. Ninad Sathe, sir, for your continuous support. Then I would like to thank our technical team from server room. And in the end, I would like to thank all the teachers, PhD students, and PG students for your patient listening. So thank you all of you. And I declare that this session is over for today.